So we last left off talking about the difference between gram-positive and gram-negative bacterial cell membranes. And I kind of boffed this one a little bit, so I apologize for that. There are going to be two main differences that lead to whether or not it's gram-negative or positive when we stain. And that's going to be the peptidoglycan. On gram-positive, it's very thick. On gram-negative, it's very thin. And so in addition, the other main difference is that the gram-negative has an outer membrane layer that is absent in gram-positive. And so those are the big differences. There are some other smaller molecular structures that are present or absent in these different types of membranes. And that would be that the gram-positive has these teichoic acid molecules that come out through the peptidoglycan, and that the gram-negative have these porin proteins to allow things to pass through that outer membrane layer. And since the gram-positive doesn't have that outer membrane layer, it doesn't have porins. So that's where I screwed up on Monday. So apologies for that. So there are some differences, and maybe a quiz question or a test question will be along those lines of, what are, compare and contrast gram-positive and gram-negative bacterial membranes. And you could talk about the peptidoglycan and how it differs. You can talk about the periplasmic space. So there's going to be more periplasmic space in a gram-negative because the peptidoglycan is thinner. You can talk about the presence of the outer membrane on gram-negative, and talk about some of the structures that are in gram-negative and not in gram-positive, and vice versa. And that's where we left off. Today, now, like I said, we'll jump into things like the gram stain, how that's done. Unfortunately, since we don't have face-to-face lab for this class, you won't be doing a gram stain, but it's a standard stain that every microbiology lab does. So we'll talk about it virtually, unfortunately, but you will be able to do it with the Labster to give you an idea. And this is going to be just, again, a table that gives you a way to compare and contrast these two different types of bacterial cell walls. Don't need to memorize the table. Okay, gram stain. The gram stain, and we talked about this briefly in the first week when we were talking about the different stains that are used as tools for microbiology. The gram stain is how we differentiate bacteria that have that cell wall with the thick peptidoglycan and no outer membrane from bacteria that have a thin peptidoglycan and an outer membrane. And we do it by using two different stains. We first stain with a crystal violet stain, which is purple, and then we wash it and rinse it, and then we use a counter stain of safranin, which stains red. So if a bacterial cell stains purple, we know it's gram-positive. If it doesn't stain purple and it instead accepts the red color, we know it's gram-negative. And then that's a really easy way to distinguish under the microscope. You just look at the cells and see what color they are. So not only is this a counter stain technique, but it is a differential stain, right? Because it says it's either one or the other type of bacteria. And we use this gram stain classification to identify all different types of bacteria, okay? It's a really, because those are the two major differences in the bacterial walls. And based on those membranes, it's going to allow certain things to pass through more easily or less easily. And so it gives us an idea of the biology of the cell based on the membrane of that cell. Okay. And so we use the gram stain again to help not only classify and put these different bacterial species into respective bins, but then also to diagnose and treat certain bacterial strains. So in the lab or in the clinic, they'll take a sample from a patient that's sick, they'll do a gram stain, and they'll see what kind of bacteria it is. And then when we go into the shapes later on, if it's a rod or if it's circular, then that will help them know where to go next for the treatment. So with the gram stain, the first step is you take your bacteria and you put it on a slide, on a glass slide. And you can kind of heat fix it to get it to stick onto the slide. And once you've done that, you add crystal violet to the bacteria on that slide. And what will happen is the crystal violet will go into that peptidoglycan layer. And as a result, it'll turn it purple. Okay. So the cell, if it's gram positive, it's going to look purple at this point because the dye, the crystals in the crystal violet stick into that thick peptidoglycan. If it's gram negative, it'll also look purple at this point because it does have peptidoglycan. And so it's going to go into that peptidoglycan of the gram negative cell as well. Now, it's not going to be as dark probably because it's a much thinner peptidoglycan. Then after you let that crystal violet penetrate into the membrane, you wash it off and you use iodine. And what the iodine does is it traps those crystals inside the peptidoglycan. Okay. So it kind of locks it in place so that when we do our next step, it won't readily release. Okay. And so they're at this point, they still look purple. They're still kind of the same. Nothing's really been done to alter it. It's at this next step of decolorizing where we add just isopropyl alcohol to the cells, that what that does is that washes the crystal violet out of that outer wall. Okay. And in this first one, because it has this outer membrane in gram negative, the iodine doesn't really get into the peptidoglycan of that gram negative membrane to fix it in place. Okay. So when this next step, when we add the alcohol, it washes out all of that crystal violet. And now it's going to lose its purple color and it's going to be clear again. Okay. So at this point, we have purple for gram negative, clear, purple for gram positive, clear for gram negative. And that's when we now do our counter stain. Okay. And the counter stain is going to go in and stain this thin peptidoglycan of the gram negative. It's not going to stain the gram positive because the gram positive is kind of full of the crystal violet and it outcompetes the safranin from getting in. Okay. And now at this point, we have our two differential phenotypes. We have the gram positive that had that initial crystal violet stain, and then we have our gram negative that's now a reddish or usually kind of a pinkish color on the slide. And that's it. It's a really simple 4-step process that gives us a lot of information about the bacteria. Now, any questions on that? before I move on.
Would you ever use alcohol or iodine after you put the saffron on?
I don't think so. Not to my knowledge, no. The iodine is to trap in the crystal violet. The alcohol is to wash away the free crystal violet that hasn't bound into the peptidoglycan. So, the crystal violet's a really um, dark, thick stain, and so it will be all over the bacterial cell, regardless of whether or not there's the peptidoglycan. And so you need that alcohol to wash it off of the cell where it hasn't bound tightly. Um, and once you get the safrano on there, you usually just rinse with water to kind of get the free stuff off, and then you're good to go. There might be some modified techniques where they use that, but the one that I've always done is that step, that process.
Sorry, can you repeat one more time why the crystal violet doesn't stay in the gram negative? Like what when it washes out? Like why does it wash out of that and not the gram positive?
It doesn't stay because the iodine helps trap the crystal violet in the peptidoglycan, but the iodine can't get through the outer membrane of the gram negative cell. So let me go back. So at this point, we have crystal violet in both of the peptidoglycan components. But then when we add the iodine, it can't get through the outer membrane of the gram negative cell to fix the crystal violet in place of the peptidoglycan. Since the gram positive doesn't have that outer membrane, the iodine gets right where it needs to be and fixes it in place. So that when we then add the alcohol, that crystal violet that was in that gram negative peptidoglycan just releases. Does that make sense?
Yes, thank you.
Mm-hmm. And I'm sure when you guys do, if you haven't already done the gram stain lab stir, that will give you a much more hands-on kind of experience and maybe some more detail and reinforce this. I know it's hard to really convey techniques and procedures using a slide deck and talking about it. It's much more educational if you do it yourself, which is what is nice about the labsters in lieu of having an actual face-to-face lab. Okay. So those are the typical cell walls. There are also these non-typical cell walls in some bacterial species. For instance, mycobacterium. So mycobacterium, think of tuberculosis and leprosy. Those are diseases from mycobacteria. And nocardia is like a pulmonary infection. And so these are technically gram positive, but they don't stain really well. Okay, so it's weakly gram positive. It's not a nice discernible purple color. And these have and the reason that they're not that purple is because they resist certain chemicals and dyes. Okay, and so that's when we use other techniques like acid fast stain, which we talked about briefly when we were talking about stains that first week. And that can be a differential stain to use to say, oh, okay, well, I can't really tell if that's gram positive or negative. I'm going to do this acid fast stain, and that will definitely work. And that will tell me, oh, okay, I must be dealing with a mycoplasm, okay, or a mycobacterium. Some bacterial cells don't even have a cell wall per se. It has a series of sterols, which are these long molecules, these cyclic rings that are used to make steroids, and cholesterols. They're alcohol rings. And so they are just a series of these molecules on the outside that, because of their structure, and the way they come together, it can be very pleomorphic. And what does that mean? That means the structure of the cell, the shape of the cell, can vary greatly from one cell to another because they don't have as organized components as do the bacteria that have cell walls. Okay, the cell walls, like we said, one of the functions is to keep its shape. And as we'll talk about later, there are very defined shapes, whether it's a rod shape or a spiral shape or a circular shape. They're pretty true to that shape. Whereas these mycoplasmas that don't have a cell wall but have just these protective sterols on the outside, it doesn't have that tight organization, and so they're much more blobby. And so blobby is a less fancy word for pleomorphic, meaning just from one cell to the next, you have pretty great variety in the potential shape. Like our cells, bacterial cells have a phospholipid bilayer that have proteins that go through them. And it is therefore a fluid mosaic model, which you guys have probably already heard of when we've talked about cell biology, right? It's a fluid mosaic model, meaning the transmembrane proteins that are embedded in that phospholipid bilayer and the different ion channels and whatnot can kind of almost move around a little bit. So it's fluid. And it's mosaic because it's not just a uniform array of the same biomolecule. It's a bunch of different things coming together. What's the purpose of the cell membrane? Well, that is not just a site for energy reactions to take place and nutrient processing and like protein synthesis. It's also where the cell says, okay, yeah, you're good for me. You're a nutrient. You're what I need. You can come in. You're a toxin. You're bad. I'm going to exclude you. So it's semi-permeable, right? It doesn't just let anything in and it doesn't just let anything out. It has fidelity on what comes in and what goes out. This is kind of like the typical cartoon of a cell membrane structure, right? You have the phospholipid bilayer here. Right, here's a phospholipid. And then here's another phospholipid here. And then their little tails are touching each other. And that's why it's a bilayer, right? Two phospholipids, one on top, one on bottom. And then in between that bilayer, or coming through that bilayer, you have different proteins. This, of course, on the bottom, this is inside the cell. On the top here, that's outside the cell. And so inside the cell, we have the cytoplasm. That's just the matrix inside, the fluid that everything is moving around in. We have the actin filaments that I talked about that allow things to transport and travel around in the cell. And then coming up through that cell membrane, we have things like proteins and ion channels and things like that to let things out, let things in, but do it in a selective manner. An integral protein is just going to be a protein that goes through the membrane and pops out. Okay, so these are all integral proteins because part of them are inside the cell, part of them are in the membrane itself, and then part of them pops out here. Going to be different from just proteins and things like glycolipids that are expressed on the outside, just the outside of the membrane. That's not an integral molecule because it's just sticking out on the surface. Okay. Any questions on that? You guys, this is probably a review for most of you because I'm sure you guys have this in like BIO 101 or 102, 103, something like that. But the point is that these bacterial cells aren't that different from our cells in that regard, right? They're different in that they don't have a nucleus, they don't have the membrane-bound organelles, but there are a lot of similarities. And why is that? That is because evolution doesn't like to reinvent the wheel. Okay, if something works, it's going to be used. If something doesn't work, then it gets tweaked, right? But you know, the similarities between bacterial cells and eukaryotic cells based on the outer cell wall is that just a lot of stuff gets recycled over time. The cell cytoplasm in our cells and in bacterial cells is very similar. It's made up of sugars, amino acids, and salts. Our cells are 70 to 80% water, just like bacterial cells, right? And that's where we get the whole concept that, you know, us as humans are about 70 to 80% water, because that's what our cells are, and we are made up of our cells, right? So there are very close comparisons between bacteria and eukaryotic cells from that standpoint, right? Because that there's that logical dissension from bacteria. Because they don't have nucleus, they have different ways of maintaining their genome. They're going to be in chromosomes, right, which are just like the tightly bound DNA. And it's usually it's a single circular strand. So just one giant strand of DNA that's all wrapped around into a nucleoid arrangement. You could also have you can also have your DNA, if you're a bacterial cell, in the form of a plasmid, which I've talked about before, which is just a circular double-stranded DNA. So it's, instead of just being kind of like rolled up into a chromosome, you have a circle. And it's interesting because these plasmids are what are used to swap genetic material between different bacterial cells, which we talked about on Monday. We talked about the pili and how the pili are used to pass genetic material from one bacterial cell to another. And so that is what they do. They transfer these plasmids. And in molecular biology, in a lab, it's the plasmids that are used for genetic engineering. And the example I used was for making insulin. If you want to make insulin, you just cut out a piece of the plasmid and you put in the gene to encode insulin. And you bind it back into that and you ligate it back into the plasmid. And then you allow the plasmid to be induced, which will then start cranking off protein from that, and whatever's there is what gets made. So, you know, and also when we were talking about Eric's topic, the first topic about potentially creating a bacteria that could break down different plastics, this would be one way that you would do that. You would take plasmid and you would splice different genes together in that plasmid and then pop that into a bacterial cell. And then that would reprogram that cell to behave in the way in which you've designated with that plasmid. Make sense? Any questions on that?
Yeah. So all bacteria cells make plasmids, or are they just like there's a set amount and they just, you know, go from cell to cell? Or I mean, how do they determine when to make plasmids, or is it just like they're just constantly making them?
Yeah, no, so I can't say whether or not, I don't think all bacteria have plasmids. It's a certain type of of genetic arrangement that is not necessarily present in every single, because bacteria is so wide and varied. There are exceptions to everything. But yes, plasmids are, so you, in a bacterial cell, you can have chromosomes or you can have plasmids. And so plasmids are pretty fairly common, you know, in terms of the types of bacteria that make them, but not standard in every single bacterial species.
So is it either or?
So is it either a chromosome or a plasmid?
Yeah.
Oh, in a cell, I would have, I that's a good question. I don't know off the top of my head. I don't know if a bacterial cell has its chromosome, and then in addition to that, it has plasmids, or if they're mutually exclusive. I want to
Yeah.
say mutually exclusive, but I'm not sure.
My question was kind of more like, how does the cell determine when to make plasmids, or because if they're constantly being transferred from cell to cell, like, is it kind of like a virus where, like, if the cell has the plasmid, a certain plasmid, then it'll just start making those plasmids, or like where did the plasmid come from? I don't know.
Yeah, so the plasmids are extra chromosomal, meaning they're produced off of that chromosome. So so I guess from that standpoint, a bacterial cell is going to have the chromosome, which is the main genetic arrangement of that cell, and then it will pop off these plasmids, and then those plasmids will then get transferred using the pilus to another bacterial cell that has its own chromosome. And then, but it's hard to say what governs that or what regulates the production of plasmids. I don't have a firm grasp of how the cell, what cues tell the cell to make plasmid and at what rate. And I don't know if it's I'm not sure if it's just a kind of a random thing that happens or if it's guided by certain factors. Is that kind of more what you were asking about?
Yeah.
Yeah, no, I know that's a very good question. I'm not sure exactly what does that, but maybe you want to look that up and share that with us at some point.
Yeah, very
But yeah, so but to just make sure we're all on the same page, the bacterial cell has the chromosome and the plasmids come off of that chromosome. And then they are used within that cell, or they can be shared to other bacterial cells. Okay. Bacteria do have ribosomes, which viruses don't, right? And this is a major component of the bacterial cell on the inside. It has lots of ribosomes, and this is what is also used to actually classify bacteria. Because if you remember that first paper we discussed in Journal Club, it was identifying all the different bacteria in the microbiome by doing ribosomal RNA sequencing, because the ribosomal RNA for each different bacterial species is unique. And the ribosomes have two subunits, a large subunit and a small subunit, which come together to then translate protein. This is where protein synthesis occurs, okay? It's going to be in all cells. That's one of the components of being a cell, is to have ribosomes, right? And so this is what they look like. You have a 70S, a 50S, and a 30S, okay? The 70S is the large and the small subunit coming together. The 50S is just the large subunit, and the 30S is just the small subunit. And does anybody know what the S stands for?
Synthesis?
No. a lot of people think it stands for subunit, but it actually stands for Svedbergs, which is a unit of measurement for sedimentation. So what happens is you spin this down in a centrifuge through some kind of media, and the rate at which it travels to the bottom of the tube, not the rate at which it travels, but the degree at which it travels, is going to be impacted by the centrifugal force and gravity. And it's going to, based on the size of that particle, it's going to travel a certain distance. And so the large subunit travels 50 Svedbergs. It travels more than the small subunit. The small subunit only travels 30 Svedbergs. Now, interestingly, when you put those two together, it only travels 70 Svedbergs. You would think if you had a 50 and a 30, that would make 80, right? But it only travels 70. And that is because when you have these two subunits together, the forces act differently than it does when they are separated. And so the overall result is that it travels only 70 Svedberg. So you don't need to know this for a test or anything, but I just like to point it out in case you're looking at this like saying, well, why is that 70 when the other two, something doesn't add up? And it's true, they don't add up. It behaves differently when they're together than when they are separate. But these are the ribosomes. They come together in these two subunits, and then through that, the RNA passes and translates into our protein. Internal structure. So we've talked about the cell wall, the components of the cell wall, and the difference in cell walls and membranes from one bacteria to another. Now let's talk about some more stuff inside. So inside the cell, what are you going to have? You're going to have ribosomes that we just mentioned. You're going to have these inclusions and these granules. Inclusions and granules are used to store things, okay? They have multiple different sizes and shapes and numbers and what they store and what they don't store, but they're used as a way to sequester certain things, sequester things that might be toxic if they're too concentrated, or to store things for a later time when nutrients are less available. Okay, so it's like the chipmunk or the squirrels stashing their acorns for the winter, right? These granules are used to store nutrients for a later time when life is more difficult and challenging. And so here's a bacterial cell, and you can see there are all these different inclusion bodies and storage granules. They have different sizes. Looks like these both have 3 of the big ones, but that's a kind of a rough idea of what they look like. Here's another really, I think this is a cool internal structure, and it's called a magnetosome. And what it is these are a special type of bacteria that can essentially precipitate out iron into these bead-like structures that are now magnetized, and they use those iron magnets for orienting themselves using the poles of the planet. So they use this like kind of like the homing pigeons as a way to navigate and get around. So I think that's so extraordinary because it shows, you know, at the most basic level, this adaptation to move around that isn't just it's not sentient by any means, but it's certainly more than just being random, right? Which is, I think, again, a very interesting, quirky, neat thing. And that's what's so great about biology, because there are all these really just radical things that organisms do to survive. So magnetosome, very cool. The cytoskeleton, that is going to be the skeleton of the cell, right? And this can be internal proteins that polymerize and depolymerize, and then help move that cell. The actin filaments, like I said, they can be used to transport things within the cell. They also give the cell its shape, as does the membrane there. But the membrane, you can think of the membrane as the skin. The actin is the bone, right? So that's why it's the cytoskeleton. We have actin, we have cytoskeletons too. So again, another parallel between bacterial cells and eukaryotic cells. Endospores. Endospores are important because they allow a bacterial cell that is in a harsh environment to basically just become dormant and survive until things get better. And if you remember, we talked about endospores. Does anybody remember why we talked about endospores and what we mentioned about them? Why they were important? Anybody? I'll give you a hint. Think sterilization.
Oh yeah, so they're the bacteria that survives at high temperatures.
Exactly. So these endospores are the things that you can't kill by just simply boiling your water. And which is why you need something like an autoclave, which has a much higher temperature than 100 degrees and has a pressurized internal component that will be harsh enough to kill these endospores. All right. So bacterial cells that have endospores, they have two different life cycles. They have a vegetative cell, which that's the active cell. That is the metabolically growing cell, right? That's undergoing different processes. The endospore is now when it's in a condition that is not bringing in nutrients, or it's very dry, and a bacterial cell, a vegetative cell, can't survive. And so then it goes into this endospore, which then allows it to just hang out, right? Nature has different ways of dealing with things, right? There are certain seeds that need to have fire in order to allow them to germinate. Endospores are a way for bacteria cells to survive when conditions are harsh. And so the process of going from a vegetative cell to creating the endospore is called sporulation. And these spores, like I said, they can withstand nasty, inhospitable environments. So in the desert, if it's too hot, or it's too dry, or it's too cold, right? It can even block off radiation and harsh chemicals. If it's in a area of high toxicity, it can make these spores, which will be impervious to the toxins. They can't get through that spore. But before that spore can then come back out of its quiescence, it has to germinate like a seed, kind of. And when it does, then it becomes vegetative again. And so this is the diagram of the life cycle, okay? And what we have is right here, we have a vegetative cell. Step one, vegetative cell says, uh-oh, it's really dry here. I'm not getting the water that I need. I need to have 70 to 80% water inside my cell. This isn't working for me. So I'm going to start step two. I'm going to start sporulation, okay? And all that is at this point, the chromosome gets duplicated, All right, so it really, the vegetative cell just has one copy of DNA, but in this process, it now makes a second copy. And then, right here, and then in the third step, it forms a septum, or a divider in between the cell right here. Okay, so it's duplicated the genome, and then now it's separating those two genomes with this septum. And so now we have what's called a forespore. And on this side here, this is the sporangium. Okay. Now, the sporangium in step 4 here, the sporangium is going to start moving its outer membrane around the forespore to engulf it. Okay, like this. And so what it does is it walls off that forespore, and now it's what's called an early spore. Okay, so it's essentially what's happened at this point is the vegetative cell has divided its genome, and then it's walled off that one of its duplicated genomes into its own compartment. Okay, and then the sporangium is going to start synthesizing these different layers. By synthesizing means it's going to start translating proteins that will then migrate to that early spore and start covering it. Okay, one of them is a cortex. This red here is a cortex layer, and then an outer coat layer here. Okay, and once that sporangium has so selflessly provided the outer coats and layers for the spore, it then starts to wither and die. Okay, kind of like humans, right? You sacrifice and you do things for your kids, and then over time you just break down and die. And that's the same thing that happens here, right? It's stressful. So you can see that the sporangium is starting to basically just break down, and it's no longer functioning, but it's put in all its energy and effort into the little spore, which now is fully completed and is ready to emerge from the residual components of that initial vegetative cell. And that brings us to step 8 here. The spore is released. And so the components of the spore is going to be the core. That's going to be where the genome is. The cortex outside in red. The spore coat in yellow. And then outside the exosporium. And that's what's going to give it its protective barrier. Okay. And it hangs out. And it's just there until something comes along and gives it a trigger that I'm in a favorable environment again. I can start germination, okay? And when it germinates, step 9, it starts releasing a new cell out of that spore. And that cell is going to have that genome in it, and then all the components of the cell membrane, and it becomes a vegetative cell. And then the process is circular. It can happen again once it goes out of those favorable conditions. Okay? Any questions on that?
Yeah.
Shoot.
Well, the book says it takes on average 10 hours for this process to happen. But you said, say that the cells start with a environment that doesn't have enough water. How do these cells just automatically start this process if they're lacking in water or nutrients or any of
Okay, so you really kind of went in and out on volume, but from what I think I understood is that you said the book says that this process takes about 10 hours, but then how long does it take, or how does the cell get the cue that the environment is not favorable? Is that what I heard, or you want to try again?
Yeah, that's what I was getting at.
Okay. Yeah, so remember, this this membrane is selectively permeable, and it's letting things in, it's letting things out, but only the things it wants in and only the things it wants out. At some point, it's going to stop receiving. It's going to sense that it is in, let's say it's in a dry environment. Okay, it's not going to be taking in any water. Now, is your question, how long does it take from initially getting that signal that it's in an unfavorable environment to then start making the spore?
Yeah, that would be part of it.
Okay. I think it varies. You know, it varies from bacterial cell to bacterial cell exactly how long it takes from getting that initial cue. It can't, it can't be too slow. It's got to start this process before the cell dies. So I think that, a matter of days, if it doesn't start making a spore, that bacterial cell will die, um, and then it's dead. So it's got to do it within a day or two. And then what was the other?
So,
Sorry.
another thing, what prevents it from dying while it's in this process? Like if there's not enough water.
I see. So in that 10-hour window when it's creating the early spore, the forespore, the early spore, and all of that, what's preventing it from dying? If that's your question. Yeah. So I mean, because it only takes 10 hours, it's walled off that genome, it's protected. And the genome itself doesn't need nutrients, right? The bacterial cell is what needs nutrients. What it's done is it's effectively just taken its genome and put it inside a safe. And that safe doesn't need to have food, it doesn't need to have water, it just needs to hang out long enough for something to happen to trigger it. Just like a seed, like I had mentioned, like, there are seeds that can get that don't survive very long. There are seeds that can survive a long period of time. There are seeds that have to have fire come in to burn off the outer coat and initiate germination, right? So because the sporulation is a relatively quick process, it can do that, and it's initiated soon enough before the cell dies that it walls off that genome and then the the sporangium breaks down. I don't know if that answered your question or not, but
Yeah, that was great.
okay, cool. Yeah, and so luckily, it's just DNA. It's DNA inside a bunch of proteins, and it just hangs out. And so that is where we're at. We're at 10:50. Any other questions? If not, you are free. I release you. But if you guys want to hang out and ask more questions, then I'm here for you. So it's up to you guys.
Yeah, so I sent you an email, Carl, of what my proposal was. And so I'm assuming that's essentially what you want me to do on the proposal assignment that's due on Friday once you release that.
Yes, I did get your email of the topic adjustment.
Mhm.
Is that what you're talking about?
Yeah, and then I went in a little bit further
Yep.
of
Yeah.
the goals of what I'm trying to do.
Yes. Yes. So for this point, at this stage, it needs to be more than just your topic, right? And that's
Yeah.
ambiguous because I don't expect everyone to be at the same stage, right? If you have your topic and what you want to do to take that topic further, then that's great. If you have it granular enough that you know the experiments or some of the experiments you want to do and what you anticipate the outcome will be, that's even better.
Okay.
If you have your questions, right, your hypotheses, that's great. Include those. So there's a bare minimum, and then there's more. And unfortunately, I'm going to grade it the same way, right? So if you have more, it benefits you because you're on the fast track, you know. But I don't want to penalize people that are trying to figure this all out. And I know how hard I've had to do these myself in real life, right? Not just theoretically. And I know how hard it is to find a topic and commit to it and think about it in this way. And so baby steps for some, giant leaps for others. It's your pace. It'll benefit you in the long run the more you provide on the outline, because again, you're going to have a head start. Okay. But when I release the assignment, I will have a few, not guidelines per se, but just like helpful tips for, formulating your outline. But yes, what you said in that email, that is good. It'd kind of be nice if it's in not just like a paragraph form, but an actual outline.
Okay.
So topic, hypothesis, experiments. But you know what? If you're not there, then that's fine. Okay.
Understand.
Okay, great.
Thank you.
Yep. Because again, all of these things are just to keep everyone on track. And so that we're not like, oh shoot, the proposal's due in a week, and I haven't done anything. So but thanks for asking that, Hava. Any other questions? Or concerns? Okay.
So I'm sorry. So you did give a few specifics there for the outline, and I know you said you're going to publish that assignment here soon. But so basically, topic, hypothesis, maybe questions we have, experiments we're proposing. Any other major milestones for us to hit in this?
No. So what I would recommend is that you look at the template for your research proposal and see what has to go into that template, and then craft your outline to answer as much of those things as you can. So if you have the significance, right? The why do I care about your project? Put that in your outline. And I think most of us can come up with a significance, right? Most of us know why it's important to treat a particular disease, or why it's important to adapt a microbe to give us some kind of service from it, a benefit for, and if you have that, add that. You don't have to, it doesn't have to be the entire significance, like, a huge, like half a page of significance. All it needs to be is a short blurb of why it's important. If you have your hypothesis, or if you have your questions, like every research project starts, it's the scientific method, right? You make an observation, you ask a question, and then based on the question, you form what you think is going to be the answer, your hypothesis. So once you have your question, that makes it so much easier to proceed. Everything else falls into place, right? When you're struggling to get your question right, you're going to struggle to design your experiments and figure out an answer to something. So for you, maybe your question is, does the gut microbiome somehow, are there certain bacterial species in the gut microbiome of female mice that are different from male mice that drive the manifestation of multiple sclerosis? I don't know if that's what it is, but like something along that line.
Yes.
And then
Okay.
that's your question, right? And so from
Mhm.
there, you can say, well, okay, if that's my question, what's my hypothesis? And your hypothesis is, yes, I think there must be bacterial species present in females that aren't present in males that are driving the MS. And then
Okay.
what's your experiment? Well, I'm going to do ribosomal RNA and sequence all the bugs in males and all the bugs in females and see if there's a difference. And then if there isn't a difference, then well, it's not that there are different bugs. Maybe it's just the way the bugs are interacting that are different, and so you go from there. But until you get that initial question, everything else is so hard. So ideally for the outline, you have your topic, you have your question, and that's what I mean by how you're going to move it forward.
Awesome. Thank you.
Mm-hmm. Any other questions? All right. Well, guys, the Labsters are available. Thank you for attending the lecture, and I'll post this as soon as I can. And I will see you Friday. So be on the lookout for the midterm review outline, and come ready on Friday if you want to with some questions or material that you'd like to go over. Okay. And until then, have a good couple days. I'll see you next time.